As blood passes through the glomerulus, about 10 to 20% of the plasma filters between the slits to be captured by the glomerular capsule and then funneled to the proximal convoluted tubule, where the fenestrae or the windows in the glomerular capillaries match the spaces between the podocytes. So this window, the fenestration or the fenestrae, these are little windows in the capillaries, just like we talked about in our blood vessels lecture, where these match up with the fenestrations or the slits in the podocytes, filtration can occur. So plasma can leave the capillaries, and so water, ions, salts, glucose, they can all leave through these slits and then cross through this basement membrane and cross into the capsular space in the podocytes, which are continuous with the glomerular capsule. These three features comprise what is known as the filtration membrane. So it's the capillary endothelium, the basement membrane, which permits rapid movement of filtrate from the capillary to the capsule, and then this third aspect, which is the actual podocyte of the glomerular capsule. So those are the three aspects of our filtration membrane. Filtering, again, based on size, filters plasma and ions, glucose, amino acids into the capsule, and it keeps blood cells, which are too large to pass through the capillaries, inside the capillaries, as well as large molecules inside the capillaries. And just to be specific, this membrane permits very rapid movement of filtrate, approximately 70 nanometers in diameter, so incredibly tiny. And so that makes up what is known as the filtration step, initiating in that renal corpuscle. And now we can look a little bit more macroscopic again and see what direction the filtrate moves. And so here is our Bowman's capsule and our glomerulus, or our renal corpuscle. And from there, it extends continuously into the proximal convoluted tubule. And so it's a convoluted tubule just because it's this convoluted nature. It's not necessarily organized. It's just this tube that's kind of crunched into this small area. So it's convoluted in the way that it looks, and it's proximal because it's close to the renal corpuscle. And so the filtrate moves through the PCT into the loop of Henle, up the loop of Henle, and then into the distal convoluted tubule. And so this then will lead into a collecting duct, which is shared by other nephrons in the system. So technically, the collecting duct is not part of the renal tubules, even though it is continuous with them. They're not for each nephron. Multiple nephrons are supplied into one collecting duct. And again, this is where the reabsorption and secretion are going to occur. So, following filtration in our renal corpuscle, reabsorption and secretion occur in these renal tubules. With up to 180 liters per day passing through the nephrons of the kidney, it's pretty obvious that most of that fluid and its contents have to get reabsorbed. That recovery occurs in the PCT, or the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule, as well as the collecting ducts. Various portions of the nephron differ in their capacity to reabsorb water and specific solutes. And so while much of the reabsorption and secretion occur passively based on concentration gradients, the amount of water that is reabsorbed or lost is very much so regulated. Most water is recovered in this proximal end of the tubule, as well as in the loop of Henle and the distal end of the tubule, but about 10% will reach the collecting ducts and then also be reabsorbed in here. And any extra water, if we're dehydrated, will be reabsorbed here, or if we're very hydrated, it'll just keep going through. And so we can see here the different substances that can be passing through. So the filtrate is this green arrow, passes through, it's plasma, a lot of water. Lots of water gets through, as well as ions. We want those back. Those are good. We need those. So water is reabsorbed into the peritubular capillaries, and they're just called peritubular because they're next to the tubes, next to the renal tubes. So they're basically the capillaries that are supplying these tubules. And so they reabsorb some of that water. So the capillaries are actually doing the reabsorption. So don't get confused here by the terminology. The tubules are secreting the sodium and the water, but really they're just moving down their concentration gradients. Sodium moves down the concentration gradient because there's less sodium in the peritubular capillaries than in the renal tubules, and then that creates a non-isotonic environment in the peritubular capillaries, so water just diffuses via osmosis to follow. And this continues throughout all of these tubules. And so what we'll see is NH3, which is a byproduct of cellular activity with proteins, will move down its concentration gradient back into the tubule so it can get secreted. And water will continue moving back into the capillary bed. And so looking at our specific renal tubules that are involved, the proximal convoluted tubule is the first point of urine formation. So that's where, right after that filtration step. So this is when we first start getting urine to be formed. It has the highest amount of substance movement in the nephron. And so secretion of sodium, water, glucose, amino acids, vitamins, all of these get moved into the peritubular capillaries because they've already been filtered from the glomerulus and the glomerular capsule, the renal corpuscle. But we want those back. And so these go back into the capillaries. So this is a reabsorption. And then significant recovery of solutes from the proximal convoluted tubule into the capillaries promotes osmosis into the capillaries. So those solutes move into the capillaries, creating a hypotonic environment in the, or actually it'd be hypertonic environment in the capillaries. So water just moves down its gradient via osmosis into the capillaries. The loop of Henle is involved in reabsorption of sodium, again promoting more water recovery into those tubes or the peritubular capillaries. The collecting duct basically picks up whatever's left over. So if we're really dehydrated, it'll reabsorb as much water as it can, and it also receives all those secretions from the peritubular capillaries that are waste products of cellular activity, such as hydrogen ions and nitrogenous wastes. So when we're looking at specific wastes, we just say that they're nitrogenous waste because they are nitrogen containing. So urine is formed in the kidneys as a product of filtration and secretion. The main secretion is that of nitrogen wastes, which are produced by the breakdown of proteins during normal metabolism. Proteins are broken down into amino acids, which in turn are deaminated by having their nitrogen groups removed. The deamination converts amino groups, or NH2 groups, into ammonia, which is highly toxic, and so cells don't want to be coming in contact with this. And so then that can further be turned into urea or uric acid by combining with an oxygen or different ions that are involved. And so ammonia, again, is extremely toxic, so most of it is very rapidly converted into urea in the liver. And human urinary wastes typically contain primarily urea with small amounts of ammonia or ammonium and very little uric acid. So that's the constitution of your urine, is usually these nitrogenous-based products of protein breakdown in your body. And so you can think of your nephron as a water treatment plant. The glomerulus is the spot where filtration occurs. The proximal convoluted tubule, we see a lot of reabsorption of ions in water. The loop of Henle is mostly involved in reabsorption of water. The distal convoluted tubule, again, reabsorption and also some secretion of ions. So these would be waste products, the secretion back into the renal tubules of wastes, as well as the collecting duct doing that with secretion of wastes and reabsorbing whatever remaining water might be needed if we're in a dehydrated state. The Micturition reflex is basically your peeing reflex. And so the urinary bladder gets full of urine, it stimulates a reflex. This green stretch receptor is a sensory receptor, and just like we talked about in our neurophysiology subject, this is a reflex arc, so it doesn't actually require brain activity, and then it sends via motor nerve to sphincters around the outside of the bladder, and this inhibits the urinary sphincters, opening it, giving you the sensation of, oh, I have to pee. And there are also higher brain centers that can be stimulating or inhibiting the external sphincter, so that we can consciously say, oh, I don't want to pee yet. I want to go to the bathroom first, so I'm going to hold it and then go. Or, all right, it's ready to go. I can pee now. And so this is just like that external sphincter that we talked about in our digestive system lecture. But that is actually all for our urinary system topic.